On va
Hello. We are going to start now. Last second to find the seat. Okay. So welcome to today's seminar in Ecology and Evolution of Montpellier, the first one of the year 2025. So as every week, it's a webinar mode. We have people on Zoom. For people on Zoom, you cannot interact with the room during the talk. But if you have questions, you can write your question in the Q&A or raise your hand and we will give you the microphone to ask your question at the end of the talk. And you can report technical issues in the chat. So now we can start with the seminar, and I have the pleasure to introduce today's talk. It's not every day that you can introduce your PhD supervisor, but you were so young when you supervised me. So.
We were still young.
We were too. I suppose everybody, Patrice, but I have to say a few words. So Patrice did nearly all his career in Montpellier with a 2-year postdoc at UCL in the UK. He's working on many things, but mostly inbreeding depression, sexual selection. He studied a lot the relationship between the genotype and the phenotype. And he's probably the best lecturer you can think about. So everybody who had him as a lecturer loved it, I'm sure. And I give you now.
Thank you very much, Nicolas. So welcome to this seminar of the year. Maybe I start with the theme, which how I liked this seminar, the last seminar of previous year. I think the best seminar ever. And I will try to do something like that, because I think what I understood from that seminar is why we are here. It's just because we have so much fun studying evolutionary biology. We can keep on for like 3 decades and we never are bored. It's just a lot of fun. And so I'm trying to do the same and try to explain to you what is it so fun to study snails and evolutionary biology in snails. So that's the title of my talk. And to tell it as a
Clique sur la diapo. Clique sur la diapo.
So yes, when I was a little kid or a teenager, I already wanted to study evolution. I found that fascinating. I read Darwin, and I told myself, well, this is really fascinating, but this old guy with the beard is boring because he's saying all the time, you're never going to observe it in a lifetime, evolution going on. It's something very old or very slow. And so when you read Darwin, you have this frustration. And I was desperately wanting to, I don't know, observe a fish, and the fish grows legs and lungs and jumps out of the water. What a fun it should be. So that was my dream. And I came to Montpellier to realize that dream, guided by a guy called Louis Thaler. And he told me, well, this dream, you can't forget about it, but I will present you a new species that you don't know, and they're really fascinating. And the new species was population geneticists, and they were traveling all over the place, and it was really fun. And they told me, okay, evolution is not what you think, it's a matter of allele frequencies that change in time. And so I said, oh, well, cool, I take that. So now I want to see allele frequencies that change in time. And that was one of my dreams, and I'm realizing it, so I will explain you how. Observing evolution in action. Another thing that evolution does is to change your way of thinking of simple things, like sex. Looks simple, but actually becomes very complicated when you speak about it with the evolutionary biologist. And so I wanted to do sex too. And another thing that evolutionary biology does is to deconstruct your sense of individuality. So you do not do sex to reproduce yourself. You do sex because your genes want to propagate themselves. And sometimes they are not in agreement with each other, and they will manipulate you in different ways to do whatever they want. So there's conflict going on within the individual, and I find this idea quite fascinating. That's the third element, which is conflict. And so what do I do to study all that? Of course, I study snails. I love snails from the first time I saw them. I think they are really a masterpiece of nature. Figs too. Snails are really cool. And so what can I do with all that? Well, ask ChatGPT to make something with all these concepts, and it failed miserably. He said, I'm meeting some difficulties. I cannot generate the image you want with conflicts, evolution, sex, and snails. And can you explore something else? Well, but we are going to explore something else then. So that's, I will tell you the last episodes of my life with snails. Starting from a very simple point, snails are not slow animals. They are the fastest animals on Earth. You don't know it, but nothing evolves faster than a snail. First. Second, all this is a matter of sex. Third, it's a matter of conflict. And fourth, then with all these ingredients, we can see evolution happening in real time. So I'm getting lots of fun with all this. So first part, snails are very fast animals. So where did I find that? Is by looking at mitochondrial DNA. So the mitochondrial DNA of snails is the fastest evolving mitochondrial DNA ever observed on Earth. And I will tell you how we discovered that. First, I present my pet snail, my favorite animal, which is Physa acuta. It's just 1 centimeter big, so it's a bit large. And it's an aquatic snail, which is hermaphroditic, simultaneous hermaphrodite. It can self-fertilize, but it prefers to cross-fertilize, so they prefer to mate together. rather than self-fertilizing, but if they are left alone, they can do self-fertilization. It lives for a few months and is a very sexually active animal. So if you put two individuals in 5 minutes, something is happening. So how this story started? It started with this colleague, Emilien, and his group in Lyon, who I managed to convince that FISA was a real nice system. And so they started working on FISA acuta and sampled them to study something completely different about plasticity and response to predation. And they sampled acuta in the region near Lyon. Acuta can be found everywhere. It's very the most widespread snail you can imagine. And they were not experienced with this species, so they started sequencing the COX-1 just to check that it was the right species they had sampled. And that's where they found this very strange COX-1 sequence, which is the D sequence that you can see here in a small tree, made with COX-1 sequences. All the normal FISA are in the green zone. Normal FISA, which is the ones they found in the populations, but also FISA that you can find in gen bank from everywhere in the world. They're all in the green zone. And this red branch here represents 10 individuals that were very divergent from the rest, but still looking really like a FISA acuta and the same. And so it was very puzzling. And they called it the FISA from space. Because it was so divergent. And we called it mitotype D later, because it was divergent. So let me show you how divergent this is. It's very spectacular. So here we have the dissimilarity in COX-1 sequence compared to one reference FISA individual, the classical FISA individual sequence in GenBank. All the FISA you can find in France, in everywhere in the world, in GenBank, they're all in this range of distance compared to the reference individuals. If you go to other FISID in the same family, the most recent common ancestor with FISA acuta would be 150 million years ago, so it's an old family. And the distance is around here. If you take sequence from a deeper clade again, you have here the hygrophila, which are all the aquatic hermaphroditic snails. And then the pulmonates, and then all the gastropods, which means back in the Paleozoic somewhere. And you see the distance between the D mitotype within FISA acuta with other mitotypes of FISA acuta is equivalent to 500 and something million years of divergence. So it's really spectacular. And it's actually unmatched by anything we know of. So we had to dig something on that. So we sequenced more. We sequenced the whole mitochondrial DNA in this 10 FISA acuta plus 10 normal individuals, the N mitotype. Plus, we found in GenBank complete mitochondrial DNA sequence from other species. And again, you see here this tree of all the mollusks we retrieved from GenBank. And here, all the FISA acuta, and here, this divergent lineage is present. I can zoom on it. So the D. So here is what it looks like. But the important thing here, so you find this very, very long branch again. What's very important is that all these colored dots are different genes in the mitochondrial DNA. All the genes, actually, there are not many. It's like 14 genes in total and 11 big genes. All these genes, they all are divergent. It's not just the COX-1 sequence we measured, it's everywhere. So it's the whole mitochondrial DNA that has gone wide and started to diverge some time ago to produce this very long branch everywhere in the mitochondrial DNA in each gene. And it's also in non-synonymous nucleotides. So It's not just synonymous changes, it's everything. And one striking thing is that when you sequence the transcriptomes of these other genes, so genes that are in the nuclear DNA, what you find is that there is no special divergence. So if you take the 10D snails compared to N snails, you have lots of divergence on the mitochondrial DNA, but on the nuclear DNA, you have absolutely no divergence. And it's true for nuclear genes expressed in the mitochondrions, the ones that produce proteins that go into the mitochondrion, and also from every other gene. So basically, it's just the mitochondrial DNA that has gone very strange. So that's where the sex comes in. So why do I think about sex when it's a matter of mitochondrial DNA? Because we know about plants, and there's other examples of mitochondrial DNAs with accelerated evolution in plants, especially in gynodioecious plants. So I need to explain gynodioecious, because Mathilde doesn't absolutely know what gynodioecious is. Finn probably doesn't either, so I need to re-explain it. Sorry. So it's the textbook example of a genetic conflict between mitochondrial and nuclear genomes. And for example, for a local example, it occurs in thyme. I never know how to say that. In which you have the normal type, which is hermaphrodite, which is coexisting with flowers in which the stamens is very reduced and they function as females. So it's a mix of females and hermaphrodites. And we call the female male sterile because actually we see it's like hermaphroditic flowers that have lost or reduced the male function. So they are sterile with regard to the male function. What is the determination of this system? It's a matter of mitochondrial DNA. And the mitochondrial, the male sterile type, the female type, is associated with some mitochondrial DNA that produce proteins that are going to change the sex of the flower and suppress the male function. So this is called cytoplasmic male sterility because it's transmitted by cytoplasmic DNA, which is the mitochondrial DNA. So we have these two types, depending on whether they have a normal mitochondrion or the CMS one. But actually, in most of these gynodioecious plants, we also have nuclear suppressors, which are genes that have evolved to neutralize the sterilizing effect of the mitochondrion. So they are called restorer of male fertility because when you have this CMS and you have no restorer, then you are male sterile. But if you have the CMS with the restorer coded by the nuclear gene, then you are hermaphroditic again. So it restores the male fertility to resist the sterilizing effect of the mitochondrial CMS. Is that okay? So it's a conflict between nuclear and mitochondrial genes, and the issue of the conflict is to decide whether you are going to become an hermaphrodite or a female. And the reason for this conflict is that these two different types of genes have a different mode of inheritance. The mitochondrial DNA is transmitted by mothers only in the ovules, not in the pollen. So it's the green part. And so this mitochondrial DNA is under strong selection to increase by any possible way the female fitness of the plant, because its own propagation depends only on the number of seeds produced. However, the nuclear genes are in a situation, they are transmitted both by pollen and ovules, so they are selected to increase both male and female fitness. So this one is selected to sacrifice male fitness if this is going to increase somehow the female fitness. And this one is selected to restore a balanced sex allocation between the two types of gametes. That's why they are in conflict, because selection has two different things to the two different genes. For a long time, we have looked for, scientists have been looking for CNRS, so this system, in animals. Why? Because, CNRS is quite widespread in plants. And we also have many animal species that are hermaphroditic. Snails, of course, some snails, but also many others, like worms, for example. And so they have been looking, why should this system not exist in animals? Actually, normally it's exactly the same thing. The mitochondrial DNA is transmitted by the mother, the nuclear genes by the two sexes. But they never found it, actually. We have very little evidence that true gynodiacy, the coexistence between hermaphrodites and females, exists in any animal species. And even in one animal species in which it has been really looked for in detail, they didn't find any, which was this worm here, the Penrose paper. So we don't have any evidence of existing CNRS in animals. We have evidence that there exists some cytoplasmic maternally transmitted elements, which are actually bacteria, symbiotic bacteria, which can manipulate sex exactly as the mitochondrial DNA can do in plants. For example, in these crustaceans, I never know the cloport, the English name. They have this Wolbachia infection that turns genetic males into females. And even the sequence from the Wolbachia that changed the sex has become in the end integrated into the nuclear genome and has become like a W chromosome, a sex-determining system. So there are cytoplasmic elements that manipulate sex in the same way as CMS, but there's no proper CMS in a hermaphroditic species because this one is separate sex. So we come back to Pfizer now. So the question was, with this strange mitochondrial DNA, are they reproductively equivalent between the normal and the very divergent mitochondrial type? And even, are they the same species? Are they compatible sexually? So what do you do when you want to know that? Emilian sent me his snails, and I used them to cross them with reference individuals that I was keeping in the laboratory, referenced by the Acuta, except that these partners were albino, which is very useful because then you can know paternity. If an albino snail lays eggs that become little pigmented snails, then the father must be a pigmented individual. So if you pair them together, if you find these ones, it means that this one has inseminated this one and got paternities. If, on the other hand, this animal does not lay eggs or lay eggs that become only white, only albino babies, then it means that this one has failed to inseminate his partner. Okay? So I fed them, lots of them, and I looked at them, their behavior. As I told you, they are very active. So you look at them and in a few minutes, something is happening. So something is happening, it can be that, which means the pigmented playing male and inseminating the albino individual. Or it can be that the pigmented plays female and is inseminated by the albino individual. And actually the time they spend doing that is heavily dependent on the mitotype. The D individuals spend practically no time in this position, while the N individuals spend more than 1/4 of their time doing that. When you look at the female role, the D and the N individual both of them spend lots of time doing female, playing the female in a population, which means that actually the D snails have a highly female biased sexual behavior. They play female all the time, instead of half of the time. And when you look at the babies, then you find that actually the D snails are practically all male sterile, which means that their male fitness, the number of babies they get from their partners, is 0. Whereas the N individuals have a positive fitness, around 40 babies on average in the experiment. When you look at their female fitness, how many eggs they lay, how many babies they get as a female, as a mother, it's equivalent. The N and D are similar. So the conclusion is that this D and N type are completely compatible with the reference FISA individual, but they cannot play, the D individuals cannot play the male role, and they have a 0 male fitness. And the reason, when you dissect them, which is what Aurel did, is that they produce practically no sperm. So they have this hermaphroditic gland in which they produce both types of gametes normally, and they store the sperm in a kind of pocket called seminal vesicle. And when you spread this seminal vesicle and look at it in the microscope, you find lots and lots of sperm in the N snails and very, very little in the D snails. So it's like sperm production has been suppressed, practically suppressed, not completely suppressed. And as a side effect, of course, because they produce very little sperm, they have trouble self-fertilizing their own eggs. So that's the normal behavior of FISA snail when it's left alone. So this is not pairing experiments, it's just leaving them alone and waiting forever for them to reproduce. The normal behavior of FISA is to wait, because FISA likes outcrossing, doesn't like to self-fertilize its eggs. And then at some point they are bored and they start laying self-fertilized eggs. So basically the normal FISA life, when it's isolated, is to stay in this blue category here, which is the ones that did not start to lay eggs yet. And at some point they start to lay egg and they go into this red category, which is the one that started to lay eggs. With time, some of them also die and go in the third category, which is the dead. Okay. When you look at what happens to D individuals, actually, they wait for ages before going into this category. So they start to lay eggs alone much, much later. And many of them die without having ever laid any egg. Means they have trouble self-fertilizing. And actually, if we zoom on these ones here, the ones that manage to lay some clutches, most of these clutches are inviable, which means that if you look here, this is the fraction, which is a few % of individuals that ended up producing at least one live offspring, which is very little for a snail. And the reason is that the clutches they lay are mostly composed of empty eggs, actually unfertilized eggs. You cannot even see the nucleus of the egg. So very, very little self-fertilization fertility. However, with François Monet, we showed that as females, they are very good. They are even slightly better than normal snails. So when you make mixed population, for example, this is a mixed population with 2 D individuals and 8 N individuals. And we make different types of mixed population. So the 2 types are in the same micro population. And this is the fertility, the number of eggs they lay. And you see that consistently the D snails lay more eggs per individual than the N snails. They are more fertile as females. They have what's called the female advantage. Something interesting is going on here. So this population is made of 9 D individuals with just 1 N individuals. For the D individuals, everything is okay. Normal fertility, equivalent to the other ones. For the N individuals, fertility is down. Why? Because there's absolutely nobody around to give them sperm. So the only way they can produce their eggs is by self-fertilizing, and they don't like that. So they delay self-fertilization and they lay less. When you are in this situation, you have 10 D individuals and absolutely no N individuals. And here there's absolutely nobody giving sperm to anybody, except a little. So we have a few leaks. It's not zero, but it's really highly limited by the absence of sperm donors around. Okay. So basically, male sterility provides a female fertility advantage, approximately 40%. Both N and D mitotypes are sperm limited when they do not have any mates that is able to give them sperm. And the D are more limited than the N because the N can at least self-fertilize in this situation, which the D cannot do. So we are in a CNS system, the same system as in plants with this mitochondrial DNA that kills the male function. And in this third part, we are going to see whether we have actually a genomic conflict, whether the male sterility is opposed by something or not, like in plants. It's an important question. Why? Because what happens if there is nothing in the nuclear DNA to prevent the action of the CMS mitochondria? This is one of the models that are typical of this scenario, which we did with Julie Roux, whose name is gone down, I don't know why, and I don't have picture of her, so I put there instead. So this kind of model, we model 2 things at the same time: the population density, which gives you the number of partners you are able to mate with in your life or to meet in your life, and the frequency of this CMS mitochondrial DNA in the population. And when you model this, you start from a situation without CMS and you introduce a CMS mutant. And it starts increasing in frequency like crazy, normal, because this the female advantage. And the only thing that prevents its complete invasion is that because at some point there are so little individuals that are male fertile that most of these male sterile individuals completely lose their eggs. They just have nothing to fertilize them. Whereas the few remaining male fertile individuals, they can self-fertilize at least. So what is the usual dynamics of this system? It's a dynamics where it increases here. Then at this stage, CMS is so frequent that most of the eggs in the population are lost. So the density of the population decreases. And one possible end is here, which is a complete suicide of the population. At the end, the density has decreased so low that everybody gets these eggs and they die. So depending on the parameters, you can have different trajectories, but basically, we have 2 types of possible endpoints in this dynamics. One is the one I told you, the suicide. The population just suicides itself, invaded by females. And another is to stabilize at a very high frequency of CMS, but which is still compatible with the renewal of the population. the population needs to produce many more eggs than actually survive than adults, but it's possible in snails, they produce lots of eggs. Which means they should have an interesting rate of increase of 9 or 8, which is quite a lot for a population. But this is just something that doesn't add up. Why? Because in this situation, so we have these two possible endpoints, suicide or high equilibrium frequency, CMS. And when we look at the population in Lyon where we found this, the frequency was actually 15%. So it's not seem like a high frequency. So we went to many other populations and looked for the D type, and we never found any population with a high frequency. All the population we found, most of them were between 0 and 3%. So you have to look at like 100 snails to find 1 or 2 that are of the D type. And the best we found was around 30%. Never more. So it's not compatible with the model. Something is missing here. So let me summarize at that stage. We found CMS for the first time in an animal. This is the first animal example. CMS prevents male reproduction and self-fertilization, but it increases female fertility. It's called the female advantage. It is associated with a very divergent mitochondrial DNA, a record breaker. There's nothing alive that has a faster mitochondrial DNA than this one. It's a mitogenome-wide evolutionary acceleration, which is due to an increase in mitochondrial mutation rates. I don't justify this, but trust me. It happens also on non-synonymous mutations or synonymous mutation equally. So it's not selection, it's really a high mutation rate. And because in these examples of fast evolution, usually it's associated with arms race, there are many examples of that. This acceleration suggests that there's an arms race going on between the mitochondrial DNA and some evolving genes that might be in conflict with that. So this is a hypothesis. It suggests that maybe we should find some nuclear restorers of male fertility, but we didn't find any for the D snails. We didn't find any restorer able to make them hermaphroditic again. So this poses a survival problem to the populations, as we saw, and it's incompatible with the data. However, we have a few leaks. So when you look at this graph, which I already showed you, we have most of the D individuals that have absolutely zero fitness. But look at this small red band here. These are just 1 or 2 individuals that have a normal fertility. So it means that even if you are a D snail, you can sometimes produce sperm and inseminate partners. It's very rare, but it occurs. So what do we do now? When you look at these snails, these little exceptions, they all belong to the same family. So they are brothers and sisters. We never know how to say sibs because it's hermaphroditic anyway. So this suggests that there's a heritable component to the penetrance of the CMS. So in some families, you are going to be less sterile than in others. So perhaps it's nuclear restorers. But if there are nuclear restorers, they must be very rare because it's only very few individuals in the populations. So we need to investigate more on this system. We want to explore mitotype diversity outside of the original Lyon population because maybe the nuclear restorers are found elsewhere in other populations. And so looking for nuclear restoration. So what we did is to design something to explore the frequency of these mitotypes and look for populations in which there will be lots of these D individuals, because that's where we have more chances to find the restorers. So we designed this PCR test. So the cool thing with this very divergent mitochondrial DNA is it's so divergent that it's very easy to make specific primers to detect it. So this is the standard test we created, which is you amplify a part of the COX-1 gene with the universal primers in black, and you add to that either a D-specific internal primer or an N-specific internal primer. So if you do this PCR with these three primers, the two black and the red, if it's a D individual, it's going to produce a short band. If it's a N individual, this is not going to work and you are going to produce a long band. Okay? And the reverse here. So if you are N individuals, you will have for this PCR a long band, for this PCR a short band, which is the case of these three individuals, and you have the reverse pattern for the D individuals. And with that, you have a very cheap way to look at many snails, kill many snails, to quantify these frequencies. But what happened actually, we looked at many snails, we found something different. In many populations, but especially in that one, the first one in which we found it, we had this pattern, which is two long bands. So it's not N or D. It doesn't match either of the two internal primers. So it's a subtype, something new. So we sequence that. And when you sequence that, it's actually a real subtype, which is called K for Kevin, who is here, Kevin Lecune. And it's really a subtype in this sense that it is very divergent from the other two, again. So again, the same story. So this is the classical mega representation with colors highlighting the difference in nucleotides. You see they're all over the place. It's just a small portion of the COX-1, but it's the same everywhere. And so we had to change our plan. So we changed the PCR test to design specific primers from the D, the N, and the K. And we put them in different positions so that we have a very short fragment for N, intermediate for K, and long for D. It gives a very simple pattern, very easy to read. So we barcoded many individuals like that. And actually, so these are French populations that we investigated. We found many of these K new type, many more than the D, actually. It was by chance that the first one we found was D, because K is more abundant. So here in different colors, you have many populations that are 100% N, normal. Many populations in purple that are a mix of N and K. Very few populations with a mix of N and D without K, and a few populations with all three types. And the frequencies are a bit like that. That's, I already showed you. The D is very rare. So every time we find it, it's like 2%, 3%, 10%. Well, but this one is generally more abundant. And we even found a few populations with like 70% or 80%. Near Perpignan, there's a population with 80% of these K individuals. The one we go for with Annick, look for snails, that's there. And if you look in GeneBank and you look for matches with the K sequence, actually, we found some. We didn't find any match with the D sequence, but the K sequence had already been sequenced and posted in GeneBank, and the people just didn't know what to do with that. So the people here said we probably sequenced the wrong species. And the people here said, we have a very new species of Pfizer. We have to protect the Snake River. It's unique in the world, blah, 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 blah. And in fact, it's just Pfizer. Okay. But the K is definitely more widespread than the D. You can find it elsewhere in the world, in all continents actually. And it's really, if you sequence the 11 big mitochondrial genes, you again find a very divergent branch for all the genes. So it's not just the COX-1, which is the K branch. And when you place it in the phylogeny, it goes into physa. So there's a little ambiguity here with another species than acuta, but this node is not supported correctly. So everything is a kind of big plateau with no discrimination about who is the real ancestor. But actually, we know it comes from physa. So it's not, this divergence is absolutely not a capture from something else very different. It's in situ, it's the production of new mutations, and it occurred twice, producing two very divergent lineage, one of which is worldwide. This was work led by Fanny Logier. Again, her name has disappeared from because of this pen, I cannot remove, but anyway, my former. Again, this suggests that the K type is probably associated with CMS like the D. So we did the same test, give them albino partners, make them mate. This is a mating with the pigmented individual playing male with the albino. And that was a surprise. The K does not behave like the D. So most of the K individuals we have are male fertile, normal hermaphrodites, just as the N individuals. So the proportion of sterile crosses is low, and it can be considered as the background error rate, so to say, failure rate. Even normal individuals fail to inseminate their mate in the time we give. We have two hypotheses now. Either this K mitotype is not CMS, it's not male sterile, it's like the N, or it is, but it's being neutralized by some nuclear restorers that are abundant in the populations we are looking. How can we disentangle these two things? We have to change the nuclear background for this K mitochondrial. We have to put them into a different nuclear background in which we know there is no restorer. So to do that, we need to introgress them. It's a very long process. So we dilute the DNA, the native DNA, into DNA coming from a naive population. This naive population is our albino population that we keep in Montpellier for 80 lab generation, no 20, no 100. And they're pure N. So probably there is no restorers in this because this has been 100 generations that they are kept without contact with the CNS. Okay. So every generation, we use a father coming from this generation to inseminate a mother from a focal line, which comes from Lyon, a K mitochondrial DNA. And we keep the babies, which inherit the K mitochondrial from their mother, but their nuclear genomes is being progressively diluted by the injection of genes from our naive population. And at the end, we end up with something like that, with the K mitochondrial into the same nuclear background as our naive population. And so what are our expectations if we have restoration in the original population? First, as we are introducing more and more genes from the naive population, the restoration will disappear and they will become male sterile, okay, progressively over the generations. Second, during this process, male sterility will be heritable, which means that the mothers that are male sterile will give daughters that are also male sterile, tend to give daughters that are also male sterile. That's the results. So we did that in parallel with K lines and N lines. The N lines remain male fertile all the time. But for the 17 generations of integration, we see the number of male sterile snails increasing and stabilizing at around two-thirds in the K mitotype. So this means that actually there are restorers in the original population where they came from. And these restorers were initially abundant, and we diluted them into non-restorer background. And in this process, we also have this heritability that I told you about, which is that male fertile mothers give more male fertile daughters than male sterile mothers, okay? So sterility is partly heritable in this system, or fertility, whatever you think about in which way. So this is because they transmit restorer genes, actually. So we have found nuclear restoration. This nuclear restoration is present in very high frequency in the natural populations where we first extracted the individuals. But something which is funny is that we don't go to 100% sterility in this system, but to something like 70 or 2/3. Which means that either in our naive population, the population kept 100 generations in the laboratory without any contact with CNRS. There are still some restorer genes, 30%, or even without restorers, the penetrance of the CMS is not full. It's something like 70%, whatever happens in any nuclear background. So we tested that. I'm going faster about this problem, but basically what we found is that, so we basically put these mitochondria into different nuclear contexts and see how it modifies the sterility. And what happens is that you have a kind of two-stage process. What happens between Lyon and Montpellier is that in Lyon, in red, you have nuclear backgrounds that nearly fully restore fertility. You have more than 80% fertility when you have this background, and the K mitochondrial. And in Montpellier, in blue, you have some variation between different genotypes, but none of them is actually restoring more than 30% fertility. So basically, all this is kind of quantitative genetic variance, but it's not a big restorer allele that changed completely the situation. So we have these two things. In Montpellier, a weak quantitative variation in the penetrance of the sterility phenotype, both for the D and the K, actually, because we did also that with the D mitotypes. And in Lyon, in the natural populations where the K come from, we have dominant restorers, which means that whenever you put them, it completely neutralizes the sterility effect of the mitochondria. And it seems to be dominant because they are here in the heterozygous state. So it's just half of the genome that comes from. Okay. And that's just a graph showing that there's absolutely no relationship between the degree of fertility in the K context and in the D context, which means that these restorers are active only on the K mitochondrial. They don't act on the D. They are not able to suppress sterility induced by the D mitotype. Okay, so we have these restorers. And now how are we going to use them to realize my dream, which is to look at allele frequencies changing in time. This is about fast evolution observed in the laboratory and a kind of model that we could call the perpetual movement. And that originates from a paper by Mathilde. But many of the gyno-DSC theory papers look alike and have this kind of similar behavior. So I'm just going to summarize this population genetic theory of gyno-DSC. This is time, and these are allele frequencies. So we start with a normal population, which is hermaphroditic. And suddenly, CMS mutant appears in the mitochondrial DNA. This CMS mutant will invade because it has this female advantage, okay? Like we observed in plants and in our snails. It invades because it has a higher female fitness than the other type. So it goes in frequency. One possibility at this point is that it goes to fixation and the population dies because it's completely composed of females. One thing that can prevent that is the appearance of nuclear restorers. So if during this process, before the population is dead, there is a restorer allele coming in the population, either by mutation or immigration, then the nuclear restorer will be strongly positively selected and increase in frequency because it has a male fertility advantage. Because the individuals carrying the restorers produce more sperm on average or more pollen than the individuals that don't carry it. Okay? Because it's transmitted by pollen, unlike these ones. So this one is raising frequency. One thing that can happen again, that it can go to fixation, and this one can go to fixation too, and then everything is back to normal. Everybody's hermaphrodite, and never talk about CMS again. Okay? But in some cases, another thing can happen is that as these ones are increasing in frequency, the CNRS mitochondria are increasingly becoming hermaphrodite again. So they lose their female fertility advantage because they're hermaphrodite, because they are with the restorers. And if, when they lose this fertility advantage, it's a little costly, then they will have a lower fitness than the non-CMS mitochondria in the same population. So this is called the cost of CMS. So it means that being a hermaphrodite, because we first sacrificed the male function and then we put it again, is less efficient than being just an hermaphrodite that didn't do anything to its male function. This cost will prevent the CMS from fixation, and CMS will start to decrease because the other type is favored. But when it does this, it can disappear completely from the population. Again, in that case, we are back to hermaphroditism again. Or what can happen is that when the frequency of these ones is sufficiently low, then the restorers start being useless because they only increase male fitness when they are with a CMS type. So if there are few CMS types, they are useless. And so if they have a cost, they will decrease in frequency again. And this can go on for lots of generations. And actually, because we observe polymorphic populations that are polymorphic both for the restorers and for the CMS, it means that we are not in these situations where we go to fixation all the time. Somehow, this model is the only one possible to maintain the polymorphism. So we expect we are going to see something like these evolutionary cycles, perpetual movement in the snails or in the plants. So in the plants, it's possible, but plants usually take 1 generation a year, so it's going to be very long to observe that. One thing with snails is that they take only 1.5 months per generation, so it's more possible to observe evolution like that. So where do we start from? Our natural populations are around here. We have very high restorer frequencies, as we observed in the, and we started from these natural populations. So what we did is to test this model. This model predicts that if you start from highly restored population, the frequency of CMS will decrease because it's being neutralized by restorers. So we make that. That's again the work of Fanny Logier, whose name is behind the band, but we start with different starting frequencies of K, mix of K and N individuals, artificial populations. But in all these populations, the frequency of restorers was the same as the original young population, which was very high. And when you look at all these populations and follow the frequency of the K mitotype over time, you see it decreasing, whether it starts from low, intermediate, or high values. In general, it decreases, and it decreases very fast. Many of these populations have completely lost the K mitotype in less than 10 generations. And if you model this on average with a wonderful maximum likelihood method by François Rousset, you see that the counter selection against the K mitotype is around 18% compared to the normal type, which is a lot. 20% less fitness, it's enormous. So there is a cost of CMS when it's being neutralized by restorers. And with this cost, the frequency of CMS decreases very fast in these populations. Now we can imagine the reverse process. The changes in frequency of the restorer value should depend on whether CMS is rare or frequent. If CMS is rare, it should increase in frequency. If CMS is rare, it should decrease in frequency. Yes, here. CMS rare, it should decrease in frequency. CMS abundant, it should increase in frequency. Okay, because the restorer is useful only when there is CMS. Otherwise, it is costly under this theory. So we do that. And so this is the first 10 generation of the experiment, which consisted in making populations either only with K or only with N type, but all of them starting with the same initial high restorer frequency. So we predict that in this population with 100% K, the restorer frequency will remain high because they are positively selected by CMS. Okay? They have this male fertility advantage in this situation. In the populations made only of normal mitotypes, the restorer should decrease in frequency because it's being costly. The problem is that when you have an N population, it's not easy to know how many restorers there is because they are all fertile, of course. So you have to inject their genes into a K individual to see if it restores the fertility or not, which is what we do. It takes several courses and it's very long, but we do it. And what you see here is that these populations, which are the 100% N without CMS at all, they have low frequency of restoration because when you inject their genes into a K context, you have high frequencies of sterility, which is the orange and yellow categories here. By contrast, if you take this population which have been maintained always in the K context, so we injected their genes into reference K individuals to do exactly the same protocol as here, and most of them are completely fertile, which is green. So in less than 10 generations, you observe divergent selection with positive, with a maintenance of very high frequency of restorer in the populations where K is abundant, and a decrease in frequency of restorers in the population where K is lacking, which again is last we predicted. So we are actually doing the 21st generation of this experiment to see if it has increased further, but more news when I finish the experiment. So I realized my dream, I saw this allele frequencies changing in time. I think it's very nice. So just to conclude, we have two mitotypes involved in CMS in this Pfizer acuta, D and K. It's the first animal example of CMS. K is more frequent than D. We have also polymorphic nuclear genes that restore male fertility that are active only on the K mitotype, but not on the D for the moment. And we have a wonderful system to observe evolution in real time and look at these genomic conflicts happening in real life, which is in my laboratory, but it's real life. So lots of perspectives, of course. We want to look for restoration in the D mitotype because we don't have it, and we still don't understand why all the populations then suicide themselves. It's thriving. Pfizer acuta is really okay. It's not suiciding itself. We want to study the change in frequency of mitotypes and nuclear restorers in the wild. So that's why we go regularly into natural population to sample these snails with Annick. We want to identify what are these restorer genes to find them in the genome. That's maybe a slightly longer process than we would like, but it will be interesting in the end. And to quantify precisely the costs and benefits of the different male sterility and restoration genotypes on male and female fitness. So all this is many things to do, and so I think I made myself clear that I find snails really fascinating. But what's fascinating also is all these people that worked with me on this project. Not all of them are in picture because I couldn't find, but I tried to put at least most of the names. Thank you very much.
The question in the room first.
Want a history from paleo history of Montpellier, and I look at CNS in Thymus vulgaris. The impression I got at the end of the study is what was determinant was really the occurrence of transient local populations, which exported more or less propagules to the next generation of populations, and that this is central in CNS, in plants, in Thymus vulgaris at least, and that this enters a whole family of models, I would say, in which you have a phase of local reproduction and then dispersal. So it could be local mate competition, it could be dominant X chromosomes and things like that.
I think there's a natural link between dispersal and I mean the female function and the male function are not equivalent with respect to dispersal in plants. I think the same is not true for animals, but I think in plants, seeds and pollen do not travel the same. And so many of the metapopulation level theory of evolution of
dinosaurs. The seeds are dispersing, not the pollen.
Yeah,
even worse in such a model with plants.
Yeah, so just one gamete disperses, and it's enough to have these founding effects and things that preserve hermaphroditism in metapopulation context, for example, prevent the invasion of. So I think the metapopulation context is really important to model these things in natural populations. So that's why, for example, even for Pfizer, I think the things that I observe in my aquaria, I completely lost CNS in some populations. This is not occurring in nature. Perhaps I completely fixed restorers in some population. This is not happening in nature. So I think the dispersal is the key to understand in foundation effects and metapopulation level things.
One thing that was important in models of CMS evolution in metapopulation is the competition between different CMS types. And I was wondering, because a lot of the mysteries about this system is you also have this coexistence between the D and K mitotypes in a lot of population, and rarely the D just by itself. So do you think that there could also be interesting interactions between those two? Competition between those two mitotypes.
There's an opportunity to for more experimental evolution with mix of DK and then the very original thing in the FISA system compared to most of the like thymus or beta or other plants is that in the plants usually you don't have any fertile type. It's only steroid types coexisting with different restorers in different frequencies. But in FISA, there is no single population we found without the normal type. Actually, it's the N type is everywhere, always. And we have sometimes like 50, 70% of K, sometimes 10, 20% of D, never all the population. Just once recently, we found one population, apparently 100% K, but it's just one population in Africa, in our freezer. I have no access to it. So I think it's, yeah, it's different. And the opportunity for competition between the K and the D type is reduced for this reason, because most of the time, the main competitor around is the N. But I think in experimental population should be cool to try and replicate what happens in the plants with steroid.
Why do you find many more population with both D and K than with just D?
It's just because K is more abundant. So they actually, it's also, yeah, okay, that's true. I didn't test whether it was random or not. Whether if you multiply the probabilities, the probability to have both is the product of one and the other. But yes, I should try that. My impression is that the D is very rare everywhere. So we have trouble even detecting it. Sometimes you type like 200 snails and you have 1 or 2 D. So it's not even easy to know for a fact that it's not there. Just there.
Thank you for the presentation. You said just at the end that your future aims are better characterize what could be the retrola.
Yeah.
Do you have any idea of what is the gene within the CMS? And if it's only it can act?
No, actually in plants, we have lots of information in that. So, but it's really not the same system. So let me tell you a little about plants, what is known about them. So in plants, usually the CMS genes are actually kind of unequal recombination events in the mitochondrial DNA that make like a chimeric new small protein. And this protein somehow, we don't really know how, inhibits the development of the pollen. And the restorers are in a family of genes that produce proteins that enter the mitochondria and modify the translation, control the translation and the maturation of the mitochondrial messenger RNA. So they enter the mitochondria and probably edit the RNA so that to neutralize the CMS. And they all belong to the same family, which is well known. And so in FISA, the only example we got in animals, it's very difficult to identify anything responsible for CMS because it's different all the place. There's no place in which the mitochondrial DNA is more different than it's different everywhere. So we will have a hard time understanding where it is. And it's not like in plants, it's not mitochondrial DNA in plants is very fat. It's full of non-coding sequence and very large, and the mitochondrial DNA in animals and in Pfizer in particular is skinny. There's absolutely no space left. So there's no chimeric genes, no neogenes, nothing easy to spot at least. So it's very difficult for us to understand what is causing CNS in this mitochondria. And for the restorers, we have no clue. But of course, we are doing a kind of genome scan to find the restorer genes. And of course, we are going to look first at the same family as in the plants, because it exists also in animals. So look at that.
Thanks a lot for the presentation. I had only one question about like the natural population. So this species is actually like spreading everywhere, and formerly it's from US, right?
Yes.
So did you plan to try to see what's going on over there in terms of, for example, the proportion of bees, etc.? Are they found everywhere?
So I already have lots of population from the US that I'm currently typing to know whether the K and D and for the moment, the situation is a bit like in France with a few K here and there, very little, absolutely no D for the moment, but I don't have so many populations that I can tell it's absent. So actually, yes, so Pfizer acuta, the origin is the western part of the United States, and probably introduced in the old world in Europe around the 17th or 18th century. And since then has a worldwide distribution. But in the process, what we know about using the rest of the genes of Pfizer acuta is that there has been no strong loss of diversity with this introduction. So the introduced, so to say, population of Pfizer acuta, the ones in the old world, in Europe and in Asia and with Africa, they are pretty much as diverse as the ones in the States. So we wouldn't expect a strong bottleneck or strong change in frequency. But perhaps there are a few more types around that we could try in the area of origin.
Do we have questions online? No? Yes, last question.
A technical question on the integration experience. How many generations do you estimate are necessary to change totally the molecular background of the image?
Actually, we said every generation you divide by 2 the proportion of the remaining genome from the original generation, so it goes very fast. So we did 17 generation, and actually I'm now at the 30th generation because I keep this line. But 5 or 6 were sufficient. You can, in the figure, you can see it. After 5 or 6 generations, we were already at the stable state. So 5 or 6 is 2 to the power of 5. It's like 100, I don't know. 32. So if you have made 5 generations of integration, the proportion of the original remaining is 1/32.
Okay, last one, because I think we are late now. Yeah, we are very late, so last one.
So you see, differently from plants, the mitotypes have many different mutations. So potentially a lot of the mitochondrial function could be affected by those mutations. So do you see, beyond main sterility?
Yes,
you have a different metabolism.
It's incredible that it still works. We agree with that. So we are trying to study that actually with mostly with Emilia, because yes, it's with that many non-synonymous mutations all over the place. It's just incredible that this just the respiratory chain still works. It's just incredible. And so we tried to measure the indices of metabolism, the respiration rate, things like that. And they didn't find any difference for the moment.
but it's still in progress, and there's perhaps something going on with temperature. Basically, the effects on male fertility and female fertility change a little between 18 and 25 degrees, and so I suspect there's something going on with metabolism and temperature and the activity of this respiratory chain, but we still don't know about it. But we are looking because it's just incredible that it still works with so many mutations.
Okay, we have to stop now. Thank you very much, Patrice.